actually visited me here. Lee, Dr. Lee came, and he did a PhD in bioinformatics, you know? So that's just the kind of person we like to see. He came here to the United States and did a 2-year postdoctoral fellowship at Ground Zero for Biomedical Informatics, which is the Vanderbilt University Medical Center. And that's actually where we first met. I came there, 5-6 years ago and gave a keynote at one of their conferences, and we had a chance to meet. And, you know, he went from there to the MD Anderson Cancer Center in Texas, and, you know, and worked as had a second great postdoc with Roel Verhoek.
Yeah.
And it really went well, and you're going to hear about that work today. And it's really been fantastic, groundbreaking work, looking at glioma, glioblastoma, multiforme. And then there's been a great collaboration here with Tom Giordano is here, and Gary Hammer. I don't see Gary. He might be in the clinic. He's a tumor board. You know, in looking at adrenocortical carcinoma, and you know, and it's absolutely fantastic. So, you know, there are 3 distinct but related disease entities. And, you know, and the goal for bioinformatics is always to try to take something that is really highly heterogeneous, is moving along in various trajectories, sometimes aggressive, sometimes less so, and trying to stratify the patients and the population so that you can understand what the best treatment would be, what the severity of the disease is from a genomic perspective. And I think that's exactly what you're going to hear today from Ziyuan in terms of genomic profile of adrenal and brain tumors. That really, I think, encapsulates it all. He's already off to a great start. There's Dr. Lawrence will be meeting with him right after lunch. There's a great dialogue with Radonk and with other members of the Cancer Center and also with the department. So welcome back. We first met again in the fall, and it's great to see you. It's wonderful to have you here, and we're looking forward to hearing what you have to say.
Thank you.
Yeah, thank you.
So, my name's Shuyuan Chen. I want to thank Dr. Ase, Dr. Lawrence, and Dr. Jun Lee for bringing here for interview, and for Dr. Tom Giordano for introducing me to the ACC project as well as the University of Michigan. So today I'm going to talk about this genomic profiling of adrenal and brain tumor. Now, I want to first give credit to TCGA, since the ACC project is part of the TCGA Rare Cancer Project, and most of my glioma project was also done within the framework of TCGA. Now, you know TCGA is a national and even international collaboration effort. University of Michigan now has become a very important part of TCGA, especially for ACC cancer. We have cases from not only from the United States, but also from Brazil, Canada, France, Germany, and Australia. So it's really international cooperation. So I'm going to talk about ACC. I'm going to talk about brain tumor. So the first part is ACC. Now, you know ACC is a rare cancer with an annual incidence around 1 out of a million. So the epidemiology is not accurate because the cancer is so rare, and this number is not even so reliable. But we see more and more adrenal cancer cases. It's an extremely aggressive tumor. The 5-year survival is less than 20%, and there's only one drug that is being approved by FDA, Mutation, which is essentially a toxic for the adrenal gland. So before we started the TCGA ACC project, there has been some efforts going on. Especially, I want to mention a few. One is from our European colleagues. There is a collaboration in Europe that's called the ANSET, the European Network for Adrenal Cancer Research. There are also some sequencing efforts from Yale and from St. Jude's Children's Hospital. So that sequences both adults and pediatric ACCs. Now, as a typical TCGA project, we have DNA and RNA sequencing, and we have methylations. We have protein arrays for part of this cohort. Now we have epigenetics for methylation arrays. Now we want to advance our understanding of this disease on the basis of what the ancestor has built and what the community has known about this disease. So if you follow the TCGA work, now if we have learned anything from this genomic profiling, so we know cancer is an extremely heterogeneous disease. Not only heterogeneous in terms of genomic changes within a single tumor, but also across the patient cohort. So a very important analysis in TCGA is we do unsupervised clustering. We want to see at different levels of genomic profiling, how do we group these patients, how do we stratify these patients, and do we find common lesions that underline these subtypes. Now for ACC, there has been some similar efforts, especially from the gene expression. Now from Tom's earlier work and from the INSET gene expression analysis, we know ACC has at least 2 subtypes. And one we call C1a, which is a very aggressive subtype. And there's another subtype we call C1b, which is much more indolent. And patient with C1b has much better survival outcome. Now, if you look at this profile, you see there are additional heterogeneity across even within C1a or C1b. So we did further refinement. We found 4 subtypes based on gene expression. And apparently from the survival curve, you see there is clinical relevance. We did microRNA expression. We did unsupervised clustering. We find 6 subtypes. This is a little crazy since we have only 79 tumors for microRNA, but we found 6 groups. What is amazing is when we look at this discriminant microRNAs, you see a big class of these microRNAs. One is on chromosome X, one is on chromosome 14q. So you basically have 2 large classes of microRNAs that can stratify your ACCs into different groups. We did DNA methylation. Now, DNA methylation separates ACC into 3 groups. From this selected CpG island, you see a lower methylated group, hypermethylated group. You have a middle group. Somehow, this lower group, low methylated group, has a much better outcome than the hypermethylated group. Now you see this hypermethylated group in all different types of cancer, in glioma, in recently profiled cholangio, in liver cancer. This seems to be a common thing in cancer. We also did DNA copy number, and this is a very, very useful analysis since from this DNA copy number you see we found 3 groups, and the top group you see. This group has a lot of copy number focal amplifications and deletions. So you see the small segments, and blue represents deletions and red represents amplifications. So small segments. Now in the second group, we see this group doesn't have too many copy number events. So this group of patients have very quiet genome. So we call this group a quiet genome, the quiet subtype. Now there is the last group that accounts for more than half of the ACC. Now in this group, you see a lot of arm level operations. Now we call this chromosomal since this group are dominated by this arm level copy number operations. Now strikingly, the noisy group has the worst outcome, and you cannot really separate the quiet from the chromosomal in terms of the outcome. So since we have this stratifications, we have the subtypes from all different types of data, but how do we integrate them? Or how do they align? So we did this integrated analysis. We call this class of clusters. So when we have multiple level separations, we tend to do this in TCGA. You will see more and more this type of analysis in TCGA papers. If you look at the pattern, you basically see all of the subtypes, they seem to converge on 3 consensus groups. You see big chunks, see big blocks on the first group, you see big blocks on the second group and these big blocks on the third group. The first group features this C1b. Remember, C1b represents patients with better outcome, and also this low methylation group. So all of this indicates this group has a better outcome. Now, when you align this to this clinical parameters, you see very few of these patients they actually die during the 3-year follow-up, and very few of them they have this tumor progression events. Now, the last this row represents the tumor progression events. In contrast, this last group features a very noisy copy number pattern and very high hypermethylation. So, here. Now, this group has the worst outcome when you look at this survival curve. When you look at this progression events, almost all of these tumors in this group they have progression within the follow-up time. And so we correlate this subtypes to mutations, to copy numbers. What we found is that the beta-catenin mutations seem to be enriched in this malignant group. Somehow, it's very few in this indolent group. Okay, so we have this. Tom, you think?
Well, you know, I'm just wondering about the one. Right. Did you look at anything further down treatment? Just in terms of long treatment? Yes, disease progression is did it occur with that window? Different cases have longer windows than others. That was really what I was asking. You know, something about the window.
And a principle in TCGA is that TCGA only profile primary tumors. So you only see recurrent tumors in a few. You only see a few recurrent glioma patients, glioma tumors in TCGA. And for melanoma, you basically have half of the melanoma tumors are metastases. But for all other tumor types, they are primary tumors. So they are untreated primary tumors. Now, what about driver mutation? This is also a huge part of TCGA. We want to identify all these putative driver events. So in TCGA, we use these tools. We use these common tools that we always use to identify driver events. So for instance, we call significantly mutated genes, use MutSeq. We call recurrent amplifications and deletions, use GISTIC. So I just show this results from our ACC analysis. We identify 5 significantly mutated genes, including p53, which probably is not surprising. We found beta-catenin, a very convergent hotspot mutation. We found menin-1, which was implicated in adrenal tumorigenesis. We found RPL22, which is a quite new gene, ribosomal protein. What is most interesting is we found a gene called PRKR1A. Now, PRKR1 is actually a regulatory protein of the pKa complex. So why this is interesting? Because a while ago, people found the catalytic subunit of the pKa complex, PRKCA, harbors recurrent hotspot mutation. This is activating mutation. And this was found in this adrenal adenomas with Cushing syndrome. So we found PRKR1 mutations, while in the adenomas you found PRKCA recurrent hotspot mutation. So given these 3 groups, given these driver events, we want to paint a landscape of ACC. Let's bring in this first panel shows the mutation density across the 3 groups. Now you can see that it seems like this group 2 and group 3, they have higher mutation frequencies or higher mutation density than group 1. So meaning these tumors have more mutations. And this is subtypes, and these 3 consensus group basically summarizes this section. When we align this driver events to the three groups, and we found, for instance, p53 mutations, a lot of p53 mutations, beta-catenin mutations in group 2 and 3, also find ZNRF3 deletions and mutations. But what is most striking is that when you look at this overall big picture, you find a lot of more mutations in group 2 and 3, which is consistent with the higher mutation density. But also remember that all of these mutations and copy number alterations we pick up here, they are all putative driver events. So they have more genomic instability. Therefore, they have more driving forces for these two groups of tumors. But when you look at the genes, and on the left, this is the mutation frequency of this driver mutation. And on the right, this is the frequency of copy number alteration of this gene. So you see, for instance, when you look at p53, for instance, now p53 is basically targeted by mutations, but not by copy number. But when you look at the second gene, the ZNRF3, which is mainly targeted by deletion, but not mutation. So there seems to be a pattern that either driven by mutation or either driven by copy number alteration. So we want to study ACC. Now we have seen the landscape of the ACC. What makes ACC special? And since ACC is a rare cancer, how do we compare ACC with other common epithelial cancers and sarcomas? We made a few attempts in comparing or doing this sort of pan-cancer analysis. Now we have, remember, TCGA typically for each cancer type would have 500 cases, but for ACC, since it's a rare cancer, we have only 91 cases. So we don't have that kind of power for every aspect of statistical analysis. So we want to put ACC in the pan-can set. We want to see, by comparing ACC, if this pan-can analysis can give us some insights. Now, one of the analyses we did is we did this mutational signature analysis. And mutation signature, the idea behind this is different types of carcinogens, they cause different mutations. So UV causes different mutations than germline naturally accumulated mutations. If the tumor has been treated by temozolomide, you will see a temozolomide signature. Now we did this mutational signature analysis, and we cluster ACC along with around 3,000 tumors. And each dot here represents basically one tumor. So this is a small diagram showing this distribution of these ACCs. What we found is a few ACCs actually cluster with colorectal cancer. And now a few ACCs actually cluster with lung cancer. So we speculate that maybe for this a few cases. Now for the cases with that cluster with colorectal, we found DNA mutations in this DNA repair gene. So they probably represent this microinstability phenotype. For lung cancer, for the cases that cluster with lung cancer, we think probably this tumor they have a smoking history. So the most insightful analysis from this pan-can is when we compare ACC, the ploidy of ACC, with more than a dozen other tumors. Now this panel shows the ploidy distribution of ACC and common cancers. What you see here is the purple represents tumors without whole genome doubling. Now, whole genome doubling is a very common phenomenon in cancer, and by this, the cancer doubles its DNA. But by looking at this ploidy, you will see ACC has a much lower ploidy than other cancers. Now, that's even true for the cases with double whole genome. So when you compare double genome, ACC also has a much lower ploidy than other cancers. Now, this ACC is only comparable to the only cancer that parallels that phenomenon is kidney lymphoma, which interestingly is also a rare cancer. So, by number, we actually have 31% of ACC that show this hypotiploid that is, ploidy less than 1.6. That's an arbitrary number, but by looking at this pattern, you clearly appreciate this pattern. So it's not surprising given that we have this lower ploidy. When you look at this copy number landscape, you see a lot of deletions. Now we see two things. Now, this is the landscape separated by subtypes. Remember, we have 3 subtypes, copy number subtypes. We have whole genome doubling and undoubled tumors. When we correct for whole genome doubling, when we look at this absolute copy number, we see a lot of DNA deletions. So that's why that explains the lower ploidy. But we also see that, for instance, when you look at the tumors in the chromosomal group, what you see is a very similar pattern between this undoubled tumors and double tumors. So we ask the question, why? Now, one thing you can infer, since if you have DNA loss, one thing that you can infer is loss of heterozygosity. So in the diploid genome, when you have a DNA loss, you see loss of heterozygosity. So we looked at arm level loss of heterozygosity, and on the left is chromosomal subtype, on the right is the noisy subtype. Remember, the pattern is most obvious in the chromosomal group. So it is true when we look at this percentage of this loss LOH event, you see it's very, very similar pattern and very, very significant correlation. That's actually even true for the noisy, even though the significance is not so dramatic. Another thing we can look at, or we can infer, is the mutation frequency. Now, we do not see a lot of homozygous mutation because it's almost impossible to have identical mutation in 2 alleles in the diploid genome. But when you lose one copy of the DNA, now you would have a lot of homozygous mutation. But what if it has a whole genome doubling? Now you can imagine that if the mutation is a homozygous mutation because of one DNA copy loss after whole genome doubling, even though you see a diploid genome, you still would see a lot. It's a homozygous mutation. But that's impossible in a undoubled diploid genome. What we see here is in this non-double group, you see in this case you have a lot of homozygous mutations. That makes sense because you have only one DNA copy. And in this whole genome doubling cases, even at the diploid genome, you see a lot of homozygous mutation. That's impossible in a normal diploid genome because you don't see 2 identical mutations. But that only can be explained by whole genome doubling. So that gives us some confidence that this is a true event in ACC. And you also see some heterozygous mutations. Now, what is this heterozygous mutation? So why they have heterozygous mutation? You can infer actually this mutation are acquired after whole genome doubling. So when you have 2 copies, you accumulate mutation, and that will be your heterozygous mutation. And this will be the subclonal mutation. So when we correlate this group of mutations, and we want to see what are these mutations, we found that p53 mutations are exclusively in this group. So meaning p53 mutation was exclusively mutated before whole genome doubling. You can imagine, that's because when you need this biallelic inactivation of p53, that's only achievable before whole genome doubling. Otherwise, after whole genome doubling, you would have to mutate both cases, or you have to use this double hit model to inactivate both alleles. So we propose that a model that can explain this is in ACC. So the ACC genome has a lot of DNA loss. Then after whole genome doubling, it doubles its DNA content. If that's true, we can infer that this whole genome doubling case would have worse outcome since that represents the late stage or the relative late stage of tumors. So when we look at this outcome data, what we found is that in the chromosomal group, indeed, the tumors that without whole genome doubling, actually patients without whole genome doubling do better than the patients with whole genome doubling. But somehow this is not true for the noisy group. Now, we do not understand why. And perhaps we have only 25 cases in this subgroup, in this noisy subgroup. Or this could be something else that can explain this. So once we establish now this whole genome doubling thing, we try to understand what happened during this process. Or the simplest way to do this is we compare whole genome doubling versus undoubled tumors. What we found is we found a few significantly mutated pathways using GSEA. One of the pathways, actually, we only found 4 pathways. The DNA repair pathway is one of these big things. And another prominent pathway is Telomere pathway. Actually, third, which encodes the enzyme part of this telomerase, is among the top differentiated genes in this whole genome doubling versus non-doubling comparison. We also see a few things, a few interesting patterns that even though ACC has a lot of DNA loss, there seems to have a few chromosomes that never lose. So for instance, chromosome 5 never, ACC do not delete their chromosome 5. Now, interestingly, TERT is situated in chromosome 5. So since we've seen this TERT, we have also seen this TERF2 amplification, which also is a very important protein that binds to telomere. So we want to do a telomere length analysis using the whole exome sequencing data. Now, nobody has ever done this. Now, somebody has developed tools to do this on this normal cell, because tumor apparently has more complex karyotype, and you have to account for tumor purity and ploidy. Now, the best data to do this is whole genome doubling, but we try using whole exome. What we found is that indeed, when we separate tumors into doubled and undoubled group, we see higher TERT expression represented by this purple bars. We correlate this TERT and TERF aberrations, including TERT promote mutations, amplifications, and TERF2 amplification. We do not find any TERF2 mutation in our data set. And we only found, as you can see, this black cells. These are, we only find 4 TERT promote mutations. But they do not seem to correlate with this whole genome doubling and or longer telomere. But we do find, what we do find is, we found that HETRAX and TERT mutant cases, they have longer telomere. If anyone knows these 2 genes, these genes have been tied with alternative lengthening of telomere, a phenotype called ALT. Now, since the discovery of recurrent LT mutations in pancreatic endocrine tumors by the Johns Hopkins group, Bert Volgassen's group, they have found that all of these mutants with HETRAX and TERT mutations, they have this ALT phenotype. But I think the most interesting observation here is not only these mutants have longer telomere, but also 6 out of 7 this HETRAX and DEX mutants, they are the non-double tumors. So this is actually a very interesting observation that, given in some paper reports that that actually telomere crisis induces tetraploidy. So I think this probably suggests longer telomere induced by this HETRAX and DEX mutation prevents tumor from whole genome doubling. In other words, this tetraploidy. Now, finally, we want to do some molecular analysis for ACC, since ACC is an endocrine tumor. It has half of the cases that are functional tumors that have hormone hypersaturation. So we select a panel of 25 genes, and this signature is highly expressed in adrenal gland only. So in other words, this genes represents how well differentiated this adrenal tumor is. So we see that a continuous, and now we have an algorithm to score this pathway, overall pathway activity. We see that a continuous increase from left to right, and accordingly, you see that many more tumors with this hypersecretion phenotype that has this higher adrenal differentiation score. In other words, the more differentiated these tumors are, they actually have more hormone hyperseparation, and they have poorer outcome. Apparently, this is very different from epithelial cancers and sarcomas, since this probably highlights the difference between ACC, a kind of functional tumor, with this common epithelial cancer. Now, finally, we come up with the complete landscape of ACC. We put all the driver events, we put these 3 groups, we put whole genome doubling events, and we put the results from our RPPA protein data analysis. We had this question that what actually is driving the group 1, since we don't see many driver events, at least from the genomic perspective. But when we compare the protein data, we actually see more phospho-PKC activity. Even though you see this white that's missing data, we only have 45 cases that has RPPA, but you still see a much higher PKC in this group. So to briefly summarize the ACC work, as a TCGA project, we present the most comprehensive genomic landscape of ACC. We use integrated analysis that separate ACC into 3 consensus subtypes. And we found ACC has lower ploidy compared to common cancer. And we think that whole genome doubling is a marker, potentially a marker for disease progression, at least in a subset of ACC. So thanks to Tom, Gary, and Dr. Roberhart for the excellent leadership in this project. And we have a lot of collaborations across the country, and also from our colleagues from Europe and Brazil, Canada, and Australia. Now, the second part of my talk, I want to talk about my analysis on brain tumor, the glioblastoma. So brain tumor is glioblastoma. Actually, brain tumor is a large collection of entities. But the most common but most deadly brain tumor is glioblastoma. So which is also the grade 4 glioma. Glioblastoma is highly heterogeneous, as its name suggests. Glioblastoma multiform. It has around 10,000 new cases per year in the U.S. And it has, the current standard of therapy is temozolomide plus radiotherapy, so-called the STOOP regimen. Now, TCGA has started the TCGA started the glioma project in 2006, and it published the first paper in Nature in 2008. But until from then, TCGA published the second landscape paper in 2013. So what we have learned from the TCGA analysis on glioblastoma is that glioblastoma at least has 4 expression-based subtypes. It has a few methylation-based subtypes, and most notably, 1 subtype that has IDH1 mutation and also exhibits hypermethylation. So we call this group a GSIMP group. All of these GSIMPs, they are proneural. They present as the expression subtype proneural. And you have a bunch of mutations and copy number alterations that correlates with these subtypes. But this is all nice results based on a large collection of primary tumors. Now, what really kills patients for glioblastoma at least is the tumor recurrence, at least for most of the cases. And for other cancers too, metastasis is the real reason that kills people. So as I said before, now glioblastoma is a heterogeneous disease. It has aggressive treatment. So you can imagine if we want to study the glioblastoma progression, we want to study the clonal architecture inside the glioblastoma. Now let's do this. So I want to introduce a few concepts, this clonal and subclonal mutation. By name, clonal mutation represents those mutations that are carried by all cancer cells. And if we believe in this one cell of origin hypothesis, then this clonal mutation should occur within that cell of origin. And subclonal mutation, on the other hand, occur in a subset of cells. So we did this clonal analysis on 250 GDM, TCGA exomes. So we separate these mutations into clonal mutations, subclonal mutations, and we did a few nice validation. So actually, in theory, there's actually no way to validate clonal or subclonal mutation unless you have a way to do single cell sequencing. That's basically not feasible. But what we did is we took different regions of one tumor and we sequenced them. Now you can imagine that the subclonal mutation should be present in all of these different regions. On the other hand, subclonal mutation probably should be only present in a subset of these regions. Now, indeed, we see a enrichment of this clonal mutation in this ubiquitous mutation that is found in multiple sectors of a tumor. We also see that a positive correlation between clonal mutation and patient age, which is also expected. So I think by these two pieces of data, we kind of convinced ourselves that this type of mutation in clonal and subclonal calls are reasonable. So we study the clonal architecture. Now we actually were not in a position to tell exactly how many clones we have in the tumor, since we use only exome data. Now, exome data doesn't give you so many mutations that you can actually tell how many clones you have. But at least we have this clonal and subclonal separation. So we sort out tumors based on how many clonal mutations, how many subclonal mutations we have. So we call this subclonality, or the fraction of subclonal mutations. We correlate with genomic alterations. We want to understand what is actually driving this subclonal mutation. What we see here is that when we correlate the percentage of subclonal mutations, we see that the cases with high subclonal mutations, they have more mutations in the p53 gene, and the same as MDM2 amplification. So MDM2 and p53, there are the two critical components in this p53 pathway. So in this high clonal mutation tumors, there are only less than 20% that has alterations in MDM2 and p53. When we look at this progression-free survival and overall survival, we found that this high subclonal cases, they actually have a better progression-free survival. But the overall survival are all the same. So in other words, now these tumors with high subclonal mutations, they tend to progress slowly, but eventually when they progress, when they reach to that point, actually it doesn't make a difference in terms of overall survival. Our result is kind of confirmed by recent analysis of a pan-cancer analysis, in which she called clones and subclones across more than a dozen cancer types. What she found in this paper is that the number of clones within that tumor actually does not always show linear correlation with the patient outcome. So when the tumor has 4 clones, the outcome is the worst. But when they have less clones or when they have more clones, actually the outcome is better. That suggests that there is a clonal-subclonal competition there, that when the tumor has more clones, it seems it progresses slowly. So remember, we did this clonal analysis in the primary tumor, but the most direct way to confirm this is you have primary and paired recurrent tumor. So we collected around 20 pairs of primary recurrent tumors. What we found is, indeed, when you separate mutations into clonal and subclonal mutations, the p53 mutants actually have a much higher subclonal frequency than than clonal mutation. So again, suggesting this p53 is really correlated with subclonal mutation. Now, so we have these 20 pairs. We want to see, at least in this case, in this pairs, how this tumor progress, how the clonal structure change. So we compare primary versus recurrent. And in this figure, this part of the figure comes from the whole genome data. You can only do this with whole genome because whole genome provides you with enough mutations. You can actually separate them into different clones. Now this part of the analysis are based on whole exome sequencing. What we found in this case is only less than 10% of the mutations they are overlapped in primary and recurrent tumor. Now this is also apparent when you look at the whole exome data, that only this part of the tumor, only part of this mutation they're overlapped between primary and recurrent. Now this is not because of the intratumor heterogeneity, because we have for the primary and recurrent tumor we have two sectors, and you can see that actually these two sectors they have a lot of overlap, suggesting we do have intratumor heterogeneity, but we also see, at least in glioblastoma, this different section of the tumor they are actually largely consistent in terms of this genetic makeup. So we built this phylogenetic tree, and this structure, the phylogenetic structure looks like this. So you have recurrent tumors, you have primary tumors that are far away from each other. And in the second case, when we compare primary recurrent, you see more than 80% of the mutations that are overlapped. Now this is also apparent in whole exome. So almost most of this primary, most of the mutations found in the primary tumor, they are also present in the recurrent tumor. But in addition, the recurrent tumor accumulates more mutations based on than the primary tumor. So when we constructed the phylogenetic tree, you see a completely different structure. This is the primary tumor. This is the recurrent tumor. You can see that the recurrent tumor largely is derived from the primary tumor. So you have some sort of linear structure here. So we propose there are at least two models in glioblastoma recurrence. So one model suggests that you have a subpopulation that is responsible for recurrence, and because the primary tumor and recurrent tumor, they're all derived from this small population of cells. And because this evolutionary bottleneck imposed by chemo and radiotherapy and surgery, so you have very different tumors, but they share limited similarities because of this small population. So presumably this represents the glioma stem cells. In the second model, so the bottleneck, the evolutionary bottleneck did not remove the primary tumor completely. So you do see there are some remaining cells in the